Hi everyone. In this lecture we're going to cover selected material from Chapter 2 of your text entitled Cognitive Neuroscience. We can define cognitive neuroscience simply as the study of the physiological basis of cognition. So this would be the physical structures that are working when any kind of cognitive act or event is happening, basically. So what areas of the brain are active, what neurons are communicating with one another, things like that. And specifically, this involves an understanding of both the nervous system as well as the individual units that comprise that system, which would be, of course, neurons. Neurons are cells that are specialized to create, receive, and transmit information in the nervous system. Each neuron has three specific parts, which are the cell body, an axon, and dendrites. Now, if we talk about how neurons might communicate with one another, we might say that neurons create a type of a nerve net, which is kind of like a highway system. And this was an older idea that basically neurons communicated continuously with each other. So this would be like if you're looking at a highway system map, and you see all of the roads intersecting with each other, but think about there not being any stoplights or stop signs there. So everything is traveling consistently along these highways. So all of this information was kind of thought to be communicated from neuron to neuron continuously without stops. Again, if you can think about it like one street would connect to another street, but again, there's no stop sign at the end of any street, so everything is just flowing continuously. However, this is contradicted now by the neuron doctrine, and this was developed by Raymond Cajal, and he used something called Golgi staining. So this was actually a technique used to stain a neuron so we could see the different parts of the neuron and how the neuron was structured and see how neurons were connected or not connected together. And what he found was that individual nerve cells transmit signals, but they're not actually continuous with other cells. And here in this figure, we can compare the first idea of the nerve net on the left side with Cajal's work with Golgi staining on the right side. And really what he did was improve Golgi staining. So as you can see, we get a much more close-up shot of what a neuron actually looks like. So here first, let's take a look at part A of this figure. So here on the left, this would be that idea again of a highway system. So you see this would be like a lot of roads here intersecting with one another, but think about there not being any stop signs here. So the idea was that all the neurons were connected together and they basically communicated information seamlessly and continuously from one to the next and on and so forth. But with Cajal's work, we were actually able to get a close-up of what a neuron looked like. And here where this arrow is pointing, this would be the neuron cell body here. And these branch-like structures would be the dendrites, and also there would be an axon here as well. But something to note is that not all the neurons are actually touching one another. They aren't connected together physically with one another. And we'll take a closer look at that here in the next few slides. So first, let's just take a look at the basic parts of each neuron. And first, which we just saw on the past slide, was the cell body, that kind of round structure. This contains the mechanisms that allow the cell to keep living. Okay, and then there are dendrites, and those are the branch-like structures that you saw. And they are reaching out from the cell body, and the function of these is to receive information from other neurons. So other information that other neurons are trying to communicate to this neuron will be received through its dendrites. And the axon is a tube-like structure filled with fluid that transmits electrical signals to other neurons. So if this neuron wants to communicate or send information to another neuron, then it will do that through an electrical signal passed through its axon. And here on this figure, we can see a diagram of what these different parts look like. Again, our round structure here is the cell body, which is holding all the stuff that keeps the cell alive. And then we have our branch-like structures, the dendrites, which again, this is what is receiving information from other neurons. And then we have our axon, which is our tube filled with fluid that sends electrical signals out to other neurons. So, how do neurons actually communicate? We've looked at the physical structures that are involved, but how does it actually occur? And we call this an action potential. And when an action potential occurs, a neuron receives some signal from the environment. So, this could be like a touch on your skin or something like that, for example. And this information travels down the axon of that particular neuron to the dendrites of another neuron. And from this, we can actually measure action potentials. We measure them using microelectrodes. So we place microelectrodes on the skin, for example, and we can pick up the electrical signal that is being output by a neuron trying to communicate with other neurons. And we place each electrode near an axon, and we can say the action potential is usually active for around about a second, so it's very fast. Now, one thing to keep in mind is that the size of an action potential is not what is measured. The size actually remains consistent from action potential to action potential. The size will never be larger or smaller. Rather, we measure the rate of firing. This is what we focus on. So, not the magnitude, but we're looking at more, again, the amount of firing. How much firing? And we say that low intensity stimuli, so something that is like a light touch, for example, will yield slower firing. Whereas if it's a very hard touch or something that is maybe even painful, will have more of a high intensity, which will yield more fast firing from the neuron. So in this figure, we can see three different examples of how we would measure action potentials using either light pressure on the skin, medium pressure on the skin, or a lot of pressure on the skin. And we can see here in portion A, when we use light pressure on the skin, we see a lower intensity rate of firing. So again, we're not measuring how large the action potentials actually are. They don't vary. What we're looking at is how often they're occurring. So all of these spaces here that you see between action potentials, this is a dead space. Essentially, there's no action potential here. So you can see that there's a fair amount of empty spaces here where there is no action potential. And here, if we're looking at B, you can see there's more action potentials occurring. And here, finally, when we're looking at high intensity firings, you can see that there's pretty consistent action potentials and there's not a lot of space between them. So this is much more high intensity firing in this case. Now, I mentioned that the neurons aren't exactly connected to one another like was originally thought with the nerve net idea. There is this space between them called a synapse. Specifically, the synapse is the space between the axon of one neuron and the dendrite or cell body of another. Now, when the action potential reaches the end of the axon, so when a neuron is communicating with another neuron and the action potential reaches the end of the axon, synaptic vesicles will open up and release chemical neurotransmitters. And these neurotransmitters, which are chemicals that affect the electrical signal of the receiving neuron, cross that synapse or gap between the two neurons and bind with the receiving dendrites of the other neuron. Now we can move on to representation in the brain. And we're going to focus on Hubel and Weisel here for a second. Now, they came up with this idea of feature detectors. Specifically, they thought that neurons basically responded best to specific stimuli. So they thought that there were neurons that we have that respond to just a specific stimulus and nothing else. And here we can see an example of Hubel and Weisel's experiments to check out whether or not we actually have feature detectors. They actually used cats. They would anesthetize them and then have them view stimuli on a screen. And they would be very simple stimuli, usually a bar, and it would be oriented in a variety of different ways. They would use vertical and horizontal and also different diagonal orientations. And they would also use stationary bars as well as moving bars. And what they did find was that certain stimuli did cause neuronal firing in the cat's visual cortex. And some of these stimuli shown here, these different bars that are shown as examples here, were ones that they did find specific neurons firing as the cat would actually view these images. So they did find some support of the idea that we might have feature detectors. Now, if we think about how we might recognize certain objects or how our neurons respond to us visually seeing certain objects, there's a couple of different ideas of how our neurons actually fire to represent a certain stimulus in our brain. and one is specificity coding. And the idea here is that the representation of a specific stimulus is really done by firing of specifically to neurons, specialized to just respond to that one stimulus and no other stimulus. Another idea is population coding. Now, this would be that representation of that same object, but the idea here is that it's done by a pattern of firing of a large number of neurons. The last idea is called sparse coding. So the idea here is that when a particular object is represented, it's done by a pattern of firing of only a small group of neurons, with the majority of the neurons remaining silent. So more of a fine difference between population and sparse coding. The only difference here is that population coding is insinuating that there's a large number of neurons that are firing. Whereas the sparse coding is suggesting that there's only a small group of neurons that are actually firing. And again, specificity coding is really more different from the other two in that it's suggesting that some neurons only fire to one specific stimulus. So again, we have three different ideas of how we might represent something in our brain. And here we'll take a look at each of the examples. First, we'll take a look at specificity coding. So this would be how we recognize faces. So the idea with specificity coding, again, is that neurons or a neuron will only fire to a specific stimulus. So here in this case, if we're trying to represent Bill, then only neuron 4 will fire and the others remain silent or do not fire. And if we're trying to represent Mary, neuron 9 will fire, but none of the others will. And then finally, Raphael will have neuron 6 fire, but no of the others will fire. So again, we have one neuron firing to represent each of these faces here, but none of the other neurons are responding. So again, the idea is that we have neurons that are specified just for one particular representation. Now, population coding is quite different in that the suggestion is all of the neurons are firing, it's just they're firing at different rates and different patterns to represent the different faces. So again, we can see regardless of the face viewed, all the neurons are firing, but they're firing in different patterns to represent the differences between the faces. And here in sparse coding, kind of similar again to population coding, in that we have a different pattern or rate of firing here, but some of the neurons aren't firing. So here for Bill, for example, we have neuron 1, 5, 6, 8, 9, 10 that are not firing, but neurons 2, 3, 4, and 7 are firing. So we have a small subset of neurons within the group that are firing to represent the face. But again, the whole group is not firing. And there isn't just one specific neuron that is responding to that particular face like specificity coding insinuates. Now, if we move on to the idea of localization of function, this is the idea that specific functions are served by very specific areas of the brain. Cognitive functioning breaks down in specific ways when these areas of the brain are damaged, and we can see some examples of that as we continue on. Now, the cerebral cortex is a very thick layer that covers the brain and contains mechanisms that we think are responsible for most of our cognitive functioning. So one item that gives us some support for this idea of localization of function is language. Now we see that language production is highly impaired by damage to Broca's area, which is located in the frontal lobe, whereas language comprehension, which is different from production, is impaired by damage to Wernicke's area, and this is located in the temporal lobe. So we have two different areas of the brain which we associate with two different cognitive abilities, and we see impairment in these abilities when there are damage, evident damage done to these specific areas. And here in this figure we can just see where Broca's area is located in the brain versus Wernicke's area in the brain. And here, before we go on to our receiving areas of the brain, we're going to actually take a look at where they are in our brain. So here we have an example of a human brain and diagrammed out to represent each of the different lobes in the cerebral cortex. And of course we have the temporal lobe in purple, which is more responsible for hearing. And then we have parietal lobe, which is more for skin senses, things like that. Our occipital lobe, of course, is responsible for vision. And then we have our frontal lobe, which is responding kind of to all of the senses and is mostly involved with higher cognitive functioning as well. And now we'll move on to talk about each of these lobes. Again, we have our primary receiving areas for the senses that we just took a look at in our figure. Our occipital lobe, of course, is vision. Parietal lobe would be touch, also temperature and pain as well. Our temporal lobe would be hearing, also taste and smell, but not as visible on the figure we just took a look at. And the coordination, finally, of all of this information from all of these different areas is happening in the frontal lobe. Now, something really interesting that we can do to provide further evidence that certain areas of the brain are responsible or correlated with certain functions is called a double dissociation. And we can do this when we have damage to one part of the brain that is causing function A, for example, to be absent, while function B is still present. And then we have damage to another area that causes function B to be absent while function A is still present. So what this does is show us that damage to one certain brain area impairs function A but not B, whereas damage to another area causes function B to be absent while function A is still there. So what this tells us then is that different functions are controlled by different parts of the brain. And we can do this through brain lesioning, but also looking at patients with brain damage as well. So how do we actually measure brain activity? Well, we can do this using an fMRI, which uses MRI technology. And here we have somebody that's actually in an fMRI, and I'm sure you guys have seen this, if nothing else, on TV before. And these are the types of images that we get. Now, specifically what we're measuring is increased blood flow in certain areas of the brain. And what we can have a person do is do a variety of tasks, maybe visual or even auditory, really any type of task, and we can measure the increased activity in certain areas of the brain. So we can kind of get a better idea of, based off of what the requirement is of the participant, what areas of the brain are more active. So that can tell us a lot more about what areas of the brain are corresponding or correlated with certain activities or cognitive functioning. Now, we have some evidence, of course, for localization of function using brain imaging. And one specific example is the fusiform face area. This is an area of the brain that responds specifically to faces. And this is located in the temporal lobe of the brain. And damage to this area causes something called prosopagnosia. And this is the inability to recognize faces. Whereas we have the parahippocampal place area, which is also called the PPA, which responds to places, so indoor or outdoor scenes, for example. And this is located also in the temporal lobe. And finally, we have the extrastriate body area, the EBA, which responds specifically to pictures of bodies and of body parts. And we've learned this using fMRI technology. Now, here would be an example of the different pictures that we would show people to measure activity in these particular areas. So if we take a look at diagram A here, this would be looking at the PPA, or the parahippocampal area. Remember, this is the area that's supposed to respond to places, but not other stimuli. And we can see the preferred areas here. When the actual area of the PPA here is lit up, we see it responding to these types of pictures, but not to these pictures here. And again, these are more scenery-based pictures, more outdoor-indoor scenes, whereas these are just a variety of different objects that aren't scene-oriented. Whereas we have the EBA, which again is geared more to responding to bodies and body parts. We see more activity in these areas here associated with the EBA, with pictures that are body part oriented. Whereas here, when shown these particular pictures, we're not seeing as many as much activity here, particularly to pictures like this, which are obviously not of any human body parts or anything like that. So this is just an example of how we can just see which areas of the brain are activated given what the person is seeing. Now moving on to distributed representation in the brain. In addition to localization of function, like we've been talking about, specific functions are processed by many different areas of the brain. Now, this might seem kind of counterintuitive to what we've been talking about thus far, but many areas of the brain may contribute to actual function. And it does appear to be a bit contradicting to localization of function, but the two concepts are actually really complementary to one another. And I'll show you an example in the next slide. So here, for an example, we have a person watching a rolling ball. Now, if we're looking at the shape of the ball, for example, we can kind of see this activated here in this particular area of the brain, whereas color would be more here, and motion here and so forth. So you see all these different portions of the whole picture being activating different portions of the brain. But in order to get the full picture here of the rolling ball, the size of the ball, the color of the ball, the shape of the ball, and the fact that the ball is actually moving, is requiring lots of different areas of the brain to work together to form the whole picture. So while each individual aspect of this whole scene is activating different areas of the brain, like localization of function would say, we do have a distributed representation, meaning that all these areas, again, need to work together to build this whole picture. We can find some support for distributed processing throughout the entire brain, looking at neural networks. Now, this is the idea that there are groups of neurons or structures that are kind of connected together or are working together to form a full picture for us. So again, we have specific areas of the brain that are responsible for specific functions, but the brain areas have to talk to one another, essentially, for us to get the full picture of what's going on around us at any given time. And these type of neural networks, we could look at them using a DTI, or diffusion tensor imaging. This can show us the kind of network of neurons that's going on throughout our brain, so we can kind of see what's netted together and kind of get some support for this idea of distributed processing, in that all the different areas are kind of communicating with one another. All right, that's all for this chapter, guys. Make sure to read the chapter carefully, and I'll get back with you next week with chapter 3. Have a great week.